And the next speaker of this important session is Mark Cooper from DuPont Pioneer of USA. And Mark will be talking about a very interesting topic, the possibilities of breeding, what can or has been changed, what can or has not. Mark, the floor is yours.
Thank you very much for the introduction, and thanks to the organizers for the invitation. I've been asked to talk about the possibilities of breeding without using any jargon. I'll try, and if I use jargon, I'll try and define that. And what I'll do is, okay, this too quickly. So I've sort of given here a bit of an outline of the way that I'm actually going to try and do this. And so one of the things that I'm going to do is build out the concept of modeling synergies. And I'm going to do that for probably a different reason for a lot of than a lot of other people put up the word modeling on here. And I think this goes to the question that Francois asked of the speakers of the first session this morning, which I really enjoyed, which really reinforced the point that the value of some of the alleles that breeders are working with and manipulating inside their breeding programs of the alleles of the genes, very context dependent. And I think one of the fundamental challenges that we've got that makes breeding for drought very difficult is that predominantly breeders are taught at universities where the assumption is made in the quantitative genetic models that we use to define selection and selection response, that the effects of these haplotypes or these alleles are static. They don't change, which is obviously a very different situation than quantifying the value of an allele within the context of some variable environments. And so we try to do that by bringing residuals and interpreting those inside the models, but it doesn't work very well. So I'm sort of trying to build that theme that I think for the future, we are going to have to build different types of models and build a toolkit of appropriate models for the questions we want to ask. And I think that in many cases, many breeders and others involved in trying to invest resources in genetic gain are not asking the right questions. So I'm sort of trying to build this theme through this talk without necessarily diving into a lot of the background detail, but hopefully give you some examples of what I mean by that. So I'm going to introduce what I think of as a foundational breeding program cycle. It's sort of very generic, and the advantage I hope with that is that it will allow you to think about, while I'm using temperate maize and breeding for that in the North America target population environments, how you can apply it to other crops. And build out perhaps a vision or a suggestion of what the state of the art could be in the future. So I'll start off with a historical perspective, talking about what we've done in the past, largely based around empirical breeding, where we're using the phenotype that we can measure in experiments to drive a lot of the decisions we make. The current perspective, where we've implemented prediction methodologies in our breeding programs because we can genotype a lot of these individuals and actually look at the alleles or the haplotypes of the regions of the chromosomes we're trying to manipulate, but we're very clumsy at this point in time in putting value on those haplotypes to drive selection decisions. But that doesn't stop us trying to make selection decisions with those regions of the genome. So we've enabled that, and I think we've moved from an estimation of trying to find these regions and describe them to a prediction paradigm. And I'll sort of try to move into the future perspective where we think about scaling prediction and starting to focus on the right questions that breeders should be asking to create the right types of products that we will need. And then also to think about how we connect that with the agronomist question that once you've created these products, thinking about how you could leverage all of that information to do a better job of placing those products. And so what I want to do is provide a suggestion that there's a real opportunity to build out an end-to-end prediction paradigm that doesn't just focus on bits and pieces of what we really need to be thinking about. Okay, so that was far too fast. Okay, there you go. So this is my generic picture of a breeding program. And so I did use a little bit of jargon, but I defined them. So MET up here, multi-environment trial. TPE, target population of environments, which just represents your target geography and all the mixture of possible types of environments that could be generated through the biophysical conditions, soil, weather, management strategies. The thing that we actually measure, the phenotype, the genotype, the genetic entity that the breeders are trying to manipulate, and E, the environment. We have a standard statistical linear model where the phenotype is decomposed into an environmental effect, a genetic effect, a residual genotype by environment interaction, and then some underlying experimental error residual. And so that basic model can be reformulated in many ways. So I got a cartoon of my breeding program here, which for a corn breeding program is just a reciprocal recurrent selection program with two heterotic groups and pedigree breeding going on inside each of those heterotic groups to develop new inbreds and new hybrids. So what we have is a germplasm pool that we're sampling from, a target population of environments that we're sampling from to construct these multi-environment trials. We do that testing. We measure the phenotypes, do a whole lot of analysis where a lot of this quantitative genetic theory and the models have been developed, predict selection response. Here's the breeder's cycle, recycling those to generate the next generation. Here's the agronomist's world. Well, you've given me something, now I have to work out how to place that with the growers in these target population environments. So I'm going to build this out with some examples. So first of all, historical perspective, I'm just going to quickly go through what has changed. And so we've run a series of experiments where we take successful hybrids from different periods of the breeding cycle. We can look at long-term genetic gain. So what you're looking at are the hybrids from the 1930s compared in the same experiments with different management that's appropriate for that time, right through to the modern period, looking at the modern hybrids, and you're looking at the long-term genetic gain. We still see that we're making this long-term genetic gain. We can actually look at this in our target geography with all the environments we encounter, but we can start to ask, how does this actually play out in drought environments? So here's a series of drought experiments that we actually ran at our managed environments for drought, and reduced nitrogen, where we are actually reducing the amount of nitrogen available to the plants and doing the same thing, and then trying to understand the yield potential, where we're looking at the long-term gain in very favorable environments. I've just highlighted the distinct period here where we transitioned from double cross breeding into single cross breeding, so multiple parents, 4 parents, down to working with just 2 parents. So what has changed? One of the things we can see is over that period of time, there's been a big change in partitioning. Just an example of this is the tassel size. The male reproductive organ has decreased in size over time. We've seen improvements in reproductive resiliency. The synchrony between the shedding of pollen and the exertion of silks has been tightened up as we've been making these improvements over time. We've reduced barrenness, so we see more plant-to-plant stability. So these all contribute to what we observe as improvements in stress tolerance. I'm sure you'll hear more about this. What else has changed? You can see if you actually look at the canopy of corn plants, the architecture's moved from very planar, flat leaf architecture to very erect leaf architecture. We've also looked at some of the functional aspects of that canopy. And here's recent work that's just in press now by Jason De Bruin that actually shows that the nitrogen content of these leaves per unit biomass has actually increased over time as well, the specific leaf nitrogen. We also have our preliminary work that's showing that the radiation use efficiency of these canopies has improved over time. In terms of water, if we compare the really old double cross hybrids with the single cross hybrids, we see very different patterns of water use over that period of time. So that we've actually changed the way in which the roots are exploring and extracting water over the full soil profile. If we look at our modern single cross hybrids, if we look at the total amount of water that's actually captured, over the whole season, there's not really any change in that. So a lot of people hypothesized that the more drought-tolerant hybrids would capture more total water. Our data do not support that. What we do instead see is that the temporal pattern of water use has been greatly shifted. And this relates to a lot of the variation that we see in the rates of transpiration under different levels of water deficit. And we've been able to fully exploit that in developing some of our targeted drought-tolerant products, our Acclimax hybrids, where we're actually conserving water during the vegetative phase, making more of that consistently available in flowering time and into the grain filling period. So these are some of the things that have changed, some of the things that haven't. So that's historical perspective where we've been selecting on phenotype, not really knowing what we're selecting for until after we look at it retrospectively. Now we're at the stage where we can take all of the inbreds that we're testing, we can actually build genetic fingerprints of those and start to describe the haplotypes, the alleles of the genes or the regions of the chromosomes. If we can actually quantify the value of those effects, one of the ways we can scale up our whole breeding effort is to actually, in parallel to the empirical program, we can actually, in computer simulation, we can run a whole virtual breeding program that's orders of magnitude bigger than your physical program using these quantifications of the haplotype effects. And that's the basis behind whole genome prediction that's actually being run today. And we've described how we've implemented that in the standard way inside our breeding programs, and enabled that in a recent review article there. But what I want to do is launch off of this and start to describe a bit of where we're at and how we're trying to move into the future and enabling this end-to-end prediction. And so really focusing on some of the questions that breeders are asking to enable them to create better products for the different target environments, and working in parallel on the agronomist's question, how can you use that information to better place these products and deal with the environment by management scenarios that make up your target population of environments. And one of the themes that I'll build out is that a link that allows you to establish that end-to-end prediction capability is investing a lot of your phenotyping efforts into building an appropriate crop growth model that allows you to predict endpoint traits that you're interested in, such as yield, as a function of the true variables that are important to you, the genetic, the environment, and the management. In this case, a lot of people write up this type of equation, and they think their job is done, but it's a nonsense equation. It's just a cartoon. And what you do is have to be explicit about the function. And I'm arguing that a lot of the physiological models embedded inside the crop growth models are appropriate functions. But what we're looking for are second-generation crop growth models that are actually designed to truly take into account the genetic variation dimension. Most of the crop models that are out there right now were developed to answer only really the agronomist type question. So they're really good at the environment and management type questions. You might do it for one genotype, but it's a very static way to think about genetic diversity, and you add some fudge factors to adjust that. So you can look at other hybrids, but that's not really a very suitable way to build this end-to-end scenario. So I'll hopefully give you some insight into what I mean by that. Before I launch, I'll sort of build that foundation out of prediction. So we know the pedigreed relationships of all of our germplasm, from founders right through to the current era of germplasm. We can actually go to a set of parents, make crosses, generate a whole population of double haploids. We create those, we fingerprint those, phenotype them in the appropriate environments. Here's some of our managed drought testing down here, and build some quantifications of the value of the haplotypes in these different types of target environments. Then use this to build out the inbred prediction scenarios and the hybrid prediction scenarios. Some summary of some of our experiences with this. This is where we're actually trying to predict new germplasm that hasn't yet been tested, using building up training data sets with previous evaluations of hybrids. And what in this example here, I've just got a summary where the lower axis, the x-axis, is prediction accuracy. This would be a prediction accuracy of 1 if you were perfectly able to predict those new genetics. This is your 0 point. So anything in here is a positive prediction accuracy. And this is just a summary from 400 different sets of germplasm that we were actually trying to predict. And this is for one of the traits we were interested in, grain moisture. So you can see that on average, we're able to get a positive prediction accuracy for grain moisture. And for grain yield, is pretty much the same picture. If you're staying within a defined target environmental situation where you're avoiding some of the real complications of genotype by environment interaction. So it's our foundation. It works. It's in place. All the breeding programs are scaling up with the capacity to run these virtual breeding programs. Once you have a particular entity, it's actually been genotyped. You've built some understanding of the traits for the different genetics you're working with. And here I've just got an example of some of the genetic variation that we actually see for some of the traits for different entities that are going through the advancement process. We can use that to parameterize some of the functions inside the crop growth model that have actually been developed to translate the environmental variability in combination with the genetic diversity to influence the traits you're interested in, such as yield. And then if we can get these inputs right, parameterize the model correctly, and get the environmental inputs right. So here, soils. So we spend a lot of time characterizing soils and understanding the soil diversity of our target environments and the managements that's used by the different growers on these soil types in combination with a lot of the weather variability. Then we can parameterize the model, and then we have a scenario to take a particular reference hybrid. So P1151 is just one of our Aquamax hybrids. And here for the target geography, I've just broken this down into a 30 kilometer by 30 kilometer grid across the whole of the Corn Belt. In each of those 30 kilometer by 30 kilometer grids, sampled out one set of soils, one weather conditions for a particular year, and one management scenario. And then we predict yield using the crop growth model for this genotype in that grid. And then we color code that. Green is higher yield, red is lower yield. And then do that for all of those 30 kilometer grids. And then you can start to do that for alternative pre-commercials that we're trying to compare to this commercial hybrid to actually look at in combination with our empirical testing, how we would predict they would perform in different geographies. So you can see an example here that in this particular year, the prediction is that this thing here, red, is not going to perform well relative to P1151. So we're at the point now where at advancement and commercialization decisions, we can actually use this type of information in combination with our empirical testing. So I want to build on this and show how we can take this further and connect these together a bit more fully. So here's an example, just taking the P1151 scenario on the previous page, take one of these particular grids. So we just looked at one scenario in this case. What we also have the ability to do in running that crop growth model from the starting point, we can actually monitor conditions throughout the season. You heard a lot about different sensor technologies. These can be deployed, and this can be used to adjust and improve and realign the crop growth model wherever we've got some of the inputs incorrect. Or it may actually tell us that we don't know something about all of the characteristics of some of these hybrids. So one of the things we can do is monitor any of these grids with a whole lot of different sensing technologies. So you heard a lot about UAV types approaches, do a lot of that type of work as well. I'm particularly interested in the opportunities for using satellite imagery. So this is just an example, if I can get it to come up, where we've actually taken a 25 kilometer by 25 kilometer view using a satellite. And we can actually get a whole lot of wavelengths currently from satellites. And there are actually a lot of satellite technologies that are coming online that give you extremely high resolution, down to a sub-meter resolution. So if there's one message that I'd make is just like we've driven the cost of SNPs to an extremely low level that's enabling us to genotype, I think satellite technology is going to make a lot of the phenotyping that we actually want to do extremely cheap in the future. We're not the only ones that are going to drive this down, and this is an advantage to us. And we can zoom in on any particular one of these grids, and we can also run experiments. where we're looking at genotype by environment by management scenarios inside those grids as well. So here's an experiment that's run inside one of these grids where we have different genetics inside here. We've got different management treatments, plant density, plant population, and we're also simulating different rainfall events at this geography by timing of irrigation. So we can look at building out the norm of reaction for any of the genetics inside this experiment, and then relate that back to the variation embedded inside the crop growth model. The other thing we can do with the satellite is we can actually zoom in with some of these high resolution satellites, so you can actually see this experiment here. You can see, if my hand was more steady, on the left-hand side here, this just coincides with the flowering stress treatment. And so it's taken at a time where you can actually start to see some of these treatment effects emerging. You can go in a little bit further, you can start to see this again, and now you can start to see some of the plot scenarios. And then from there, we can actually start to extract appropriate wavelengths to look at some of the time course characteristics that help us to interpret the genetic diversity that we're expressing inside these experiments. So this is, we're sort of coming to the point where phenotyping is becoming a real opportunity to scale. This in no way substitutes for the necessity of on-the-ground ground truthing. And here's just an example of some of the work in these types of experiments. And if there's one message I'd sort of leave you with in this, is that purposeful phenotyping rather than just phenotyping is really important. So as well as investing in these sensing technologies, really investing in how we do the right crop physiology and agronomy is just as critical. Doesn't replace it. So here's the scenario we're at. Now I want to move on to this end-to-end prediction and start thinking about combining these questions. The way to think about this is tying back to some of the scenarios before and the diversity of environments we're trying to sample. Our multi-environment trials are just really some sample of this target population of environments. The genetics we're dealing with is just some sample of the possible genetics we could create. The environments we're dealing with, whether they're by management or biophysical sampling, are just a subsample of our target population environments. Here's your two levels of prediction, and out here are the agronomist questions, around here are the breeder's questions. How can we actually tie these together? So one of the ways we've been looking at doing this is actually taking our whole genome prediction methodology and embedding inside the likelihood algorithms the crop growth model in place of the quantitative genetics infinitesimal model, which is the core model that's actually used for the static quantitative genetics model for the classical whole genome prediction that's out there. So here's a theoretical example of where we implemented this. And in this case, we're looking at the prediction accuracy for scenarios here on both axes. This is one of the standard prediction methods. And then comparing that to the crop growth model whole genome prediction. This is the one-to-one relationship. Whenever you've got a point on that line, they're doing equally as good. Whenever a point goes above the line, the crop growth model is able to actually adjust these effects correctly so that the whole genome prediction does a better job of predicting the relative performance of new genotypes in these new environments. So this is sort of an opportunity to actually embed a lot of what we've been doing on the physiology, the phenotyping, on the agronomy type work inside the actual breeding program and the breeders' questions. We've taken that theoretical work and we've actually implemented it so that we can do it empirically. So it's all driven now with our real data. And so just as an example of some of the key components, we're ultimately trying to get better estimates of these parameters that are actually looking at the norm of reaction of the haplotype effects using the crop growth model in combination with phenotyping focused on defining the priors, building the crop growth model, and then actually building training data sets in some of our targeted and phenotyping environments. So this is an example where we're doing this for drought, and then we can actually build high quality training data sets. One of the key points here is we're actually tying the effects of these QTL to the functions and the traits inside the crop growth model that are actually less dynamically responsive or unpredictably responsive to a lot of the environmental variables. So some of these traits that are much more predictable to these environmental inputs, such as leaf number or flowering time tied to temperature and so forth. We tie the effects to those traits and then use the crop growth model to do the yield prediction. And then here's an example of putting that in place and just applying it to some new populations that we were trying to actually predict in that case. So now I want to just move on to the final point that I'd want to cover, and that is, so we've got our classical multi-environment trial. In this case, we ran an experiment where we manipulated water availability. So yield is a function of the genetic variation. We got two classical types of hybrids here, different environments, different water availability, management plant population to shift the supply-demand ratios. It's all conditional on water. And here are two genetics that I'll spend a bit of time looking at. One that's sort of more of a high potential hybrid that can perform well in these very favorable environments, one that's actually a bit more stable. This is sort of a good description, but what I want to do is actually translate this into something that's more tied to the biophysical relationships inside that environment. So in this case, because we know water is a driving variable, we can actually quantify all of our environments in terms of the amount of evapotranspiration that was accessible through the crop and the environmental conditions in those scenarios. Look at that yield front, the potential yield. You can look at any particular scenario and say, how close are you to the potential with the management scenario for that genetics? And you can actually look at different genetics also in terms of different fronts inside this framework. So now I'm just going to take this experiment and place this inside a TPE view using this type of framework here. And so first of all, these two hybrids, we know some of the characteristics that differ, and we can quantify their effects and tie those to some of the underlying regions. So canopy size of the most stable hybrids, a bit smaller. We've got some elevated level of restricted transpiration. There's improved reproductive resiliency, whereas the potential hybrid is sort of contrasted to that. And so this is a scenario now where I've taken that 30 kilometer by 30 kilometer grid, taken both of those hybrids, taken one soil situation, one environmental situation, and one management situation, done that for 60 years across the whole of the Corn Belt. There's something like 360,000 yield predictions here. And what you can start to do is look at the potential yield front for the different genetics, and then you can start to contrast your genetics in this picture. And what you can start to do is look at the difference in the opportunities for a much wider range of water scenarios for the stable hybrid, the potential hybrid. There's a lot of things I could talk about here. One thing I'll point out, if you look across this front here, you can see there are both red and blue dots, which means even the more high potential hybrid that has some weaknesses, if you get the management right and you protect it at flowering time, you can actually get the same yield as the more stable hybrid in that particular environment where you've got some level of water available. The risk you run is if you don't get the management right, you've got a higher probability of dropping yield off in that particular environment. So this is some of your genotype by environment by management interaction, fully interpreted through the genetics and the integration with inside the crop growth model. And you can also see the difference between the yield potential hybrid. The stable hybrid has some trade-offs with that reproductive resiliency in terms of the total number of kernels that can actually be set. What I want to now do is take that experiment that I just showed you, and we can actually quantify the evapotranspiration in each of those scenarios for the different genetics, and then plot the yield relative to the ET for those genetics inside those experiments. And you can see that our experiments reside inside this total possible space, and you've now got some frame of reference for starting to think about your prediction problems and the limits of predictability that you're actually working with in this G by E by N space. We can also reinterpret our norms of reaction in this case. The nice thing about this is you can start to demystify some of these mystical crossover interactions, and you can see that in this case for these hybrids, this crossover interaction that you see statistically in our real data is really a coincidence in this experiment of the point at which the more stable hybrid is reaching its yield potential, no matter how much more water you actually put in there, whereas the yield potential, the hybrid, has the ability to keep going. So you've got a biophysical interpretation now of your G by E by M scenarios. So quickly, in conclusions, hopefully what I did do was set the scene for a lot of the other talks you're going to hear where people are actually going to have the luxury of talking about some of their particular challenges that they're working on, that sort of think about that within this overall framework and opportunities. Historically, we selected on phenotypes. There's a lot of people still working in this space, a lot of investment in how to correctly design and analyze and phenotype for multi-environment trials. And we can describe the changes that have happened and try and interpret those. There's a lot of work in that area. Current perspective of where we're at and where I'd say any breeding program has an opportunity is that with the lower cost of genotyping, you can start to integrate molecular technologies inside breeding programs and enable commercial scale or target scale prediction. The future perspective, I think, is if we can actually build a proper toolkit of the appropriate models at the level of the question that you're trying to answer for your target geography and the genetic diversity that you actually have access to enable this end-to-end prediction rather than just predicting pieces of the overall problem that we have. I think we're at the point where we have an opportunity to really fully integrate the product creation and the product placement issues and answer breeders' questions and agronomist questions to ultimately deal with the growers' questions using the toolkit that we now have the opportunity to build. So anyway, thank you for being patient and listening to me through the tour of trying to build out this picture and set the scene of the possibilities of breeding with a historical, current, and a vision of where perhaps the future perspective could be. Thank you very much.
Right. Thank you, Mark. Thank you for the excellent and exciting presentation.